Namaste students, my name is Dr. Niyaz Dholia, Faculty of Agriculture and Veterinary Science, Jayoti Vidyapeeth, Women's University, Jaipur. So today we are going to discuss a practical session, that is PCR, the polymerase chain reaction, and this belongs to the subject medical biotechnology lab. So what is PCR and how to perform that? So basically the PCR is a polymerase chain reaction, and it is a method that analyzes a short sequence of the DNA or the RNA, and even in sample containing only minute quantities, means a very less amount of DNA or RNA can be identified using the PCR technique, which is not possible in other techniques. So the basic funda is that the PCR amplifies the sample, means the gene of interest you want to know or you want to analyze, that is first amplified. So the previously amplification of the DNA involved the cloning the segment of interest into vectors for expression in the bacteria and took weeks. But now we PCR done in test tubes. We amplify the DNA in the test tubes, and this is the highly efficient in that untold numbers of copies can be made of the genes. So PCR use the same molecule that nature uses for the copying DNA, that is the DNA polymerase, and machinery is also same. So for amplification, we need two primers. One is the forward primer, one is the reverse primer. That are the short single-stranded DNA sequences that are synthesized or correspond to the beginning and the ending of the DNA stretch to be copied. Then the DNA polymerase, the enzymes that moves along this segment of DNA and reading its code and assembling a copy. Then we need the nucleotides, or we can say the building blocks of the gene. So they are building blocks that the polymerase need to make that copy. So how it is performed? So in the PCR, the three major steps are involved in the polymerase chain reaction. These steps are repeated for 30 to 40 cycles. So these cycles are done on an automated cycler, that is the thermal cycler, a device that rapidly heats and cools the test tubes containing the reaction mixture. So in each step, there are few sub-steps are there, that is the denaturation, annealing, and the extension. In the denaturation, we alter the alteration of the structure. The DNA will denature in this state. And in the annealing step, this is the joining step, and the extension that takes place at the different temperatures. So denaturation it occurs at around 94 degrees Celsius, and the temperatures we can change we can do the minute changes in the temperature according to the samples, according to the requirement. Generally, the denaturation occurs performed at 94 degrees Celsius, or if in the Fahrenheit, we can say the 201.2 Fahrenheit. So in the denaturation, the sample, the double-stranded DNA that denatures and opens into the two single-stranded DNAs. In the annealing, it happens at the medium temperature, that is around 54 degrees Celsius, and the primers are annealed here. Anneal matlab, anneal means they bind to the template DNA. So the primers pair up or anneal with the single-stranded DNA templates. The template is that the stretch of the DNA to be copied. So on the small length of double-stranded DNA, the polymerase attaches that it starts the copying the template. Then in the extension, it happens at 72 degrees Celsius, and if in the Fahrenheit, we can say the 161.6 degree Fahrenheit. So the polymerase works the best, and the polymerase and the DNA building blocks complementary to the template are coupled to the primer and making a double-stranded DNA molecule. So extension is the main polymerization reaction. Here the DNA polymerase enzymes work, and the DNA polymerase may be of different sources. It may be from the bacteria, it may be from the phases, and the sources may be others. So with one cycle, a single segment of double-stranded DNA template is amplified into two. Okay, so when one cycle is completed, means the single-stranded segment of the double-stranded DNA template is amplified into two separate pieces of the double-stranded DNA. So this is like the application. Okay. So these two pieces are then available for the amplification in the next cycle. Again, when the second cycle starts, a denaturation step will start. These two DNA templates, double-stranded DNA templates, they denature and they convert into 4 single-stranded DNAs. So in this step, we have the 4 templates. Okay. So as the cycles are repeated, more and more copies are generated, and the number of copies of the template is increased exponentially. Okay, so exponential increase happens here, and we get the ample of the DNA molecules, ample of the genes which we want to detect. Okay, so in the initial sample, the gene of interest which you want to detect was present, but that is not present in the detectable range. So the PCR is very helpful to detect the samples which are present in very low amount or very lesser amount is there. So this was all about the PCR. So in this we have studied that what is the PCR, the definition. It is a polymerase chain reaction, and it is used to analyze the presence of a gene or DNA or a short sequence of the nucleotides. Okay, so in this we need various things, that we need a template, we need the primers, forward primers and reverse primers. Then we need the enzymes, different enzymes, and we need building blocks, that is the oligonucleotides. Then we need a buffer, because a medium is required to react the biomolecules together. So that is a buffer. Then we need ions. Some ions are necessary for the reaction. So the all materials together are known as a reaction mixture, PCR reaction mixture. And the PCR machine is generally, it is named as a thermal cycler, because it set or it fixed the temperature. It cools very in a very short time, and the temperature increases in very short time. So a cycle of a set parameters for the temperature and time is fixed in that machine. So that is known as the thermal cycler. Okay, so in the PCR we study that you have to repeat the cycles at least for 25 times, or 25 to 40 cycles you can set according to the requirement. Okay, and the steps which are performed in the one cycle is the denaturation, then annealing, then extension. Okay, so what happens in the denaturation? The double stranded DNA melts and denature, and it opens into two pieces of the single stranded DNA. And what happens in the annealing? So the primers, okay, which you use as a forward primer or the reverse primers, they anneal to their complementary sites. Okay, so because if you have a DNA or a length of a DNA, but you do not want to amplify the whole DNA, you need only one gene, okay, that is of your interest. So you prepare or you make the primers for that particular gene, and the cycle, the polymerization will occur for that particular gene only. That will not amplify the whole DNA. Okay, so that's why you save the materials, you save the time for the polymerization. So that was all about the PCR or polymerase chain reaction. So hope you are satisfied with this detailed session. If you have any doubt, any query, you can write in the comment box. And this session is powered by Digital Version 2, Jayoti Vidyapeeth, Women's University, Jaipur. Thank you.